Sunday Chai and Wine. Today is fifth Sunday, and as a part of our fifth Sunday Chai and Wine, today is up close with science, where we visit a lab in TIFR and bring the science pursued by the lab to you. So today we are visiting a laboratory of Professor Sundar Ram Naganathan at the Department of Biological Sciences here, and they probe very interesting things in biology, which is like why is it that there is a symmetry in vertebrates along left to right. So Dr. Sundar joined TIFR 4 years ago.
Yeah, 2.5 years ago.
Just 2.5 years ago. And before joining TIFR, he completed his doctoral studies in Germany and postdoctoral stints in the western part of the world. And then he has set up his laboratory in TIFR. And I trust today yourself and your students will be explaining.
Thank you very much, Ullas, for the kind introduction, as well as to Arnav and Ullas for requesting us to be part of this Up Close with Science. It's an exciting opportunity to show you all. So first of all, welcome to Up Close with Science. And Sundar, and this is Sweta, a fourth year PhD student, or almost a fourth year PhD student. And this is Subhanjan, who's a second year PhD student, who will take over, and show everything about the lab, what we do, what we are interested in, the kind of problems that we work on, the model system that we are looking into, and so on. But maybe briefly, I'll give you a couple of sentences on what we do before they fully take over, right? So our lab is called Symmetry Lab, and the reason for that is we are particularly interested in understanding how a left-right symmetric body plan emerges during embryonic development. Okay, so there's a lot of jargon here. So let me deconstruct a little bit for you, okay? So if you take your own body, this they will also explain a little bit more in detail, but I'm giving you a brief overview. If you take your own body, you can draw three body axes, right? What do I mean by body axis? You have one sort of line that you can draw from the head towards the leg. Okay, so that's a vertical line that you can draw. You can draw one line that goes from the back of your body to the front. That's a horizontal line, which is perpendicular to the initial line that you have drawn. And there's another line that you can draw between the left, that goes from the left to the right or the right to the left. So that is another line, right? So these three body axes are essentially perpendicular to each other. Okay. Of these body axes, the left-right axis is what we are interested in. Why? Left-right is a very peculiar or a arbitrary axis in a way, in the sense that for me, left is this side. For you as viewers, for you, the left will be the other side. So why is it flipped? Right? So that's a easy question to ask, right? And the reason for that is my front-back axis is pointing towards you, and your front-back axis is pointing towards me. Because you're flipping the front-back axis between the two of us, the left-right axis automatically gets flipped. So for me, left goes that side, for you, left goes that side. So that's why left-right axis is an arbitrary axis, and it really depends on how you define the head-leg axis and the front-back axis. And what you call as left and what you call as right is entirely dependent on that. Okay. The second reason why we are interested in looking at left-right body axis is if you look externally, everything is very symmetric, right? Your eyes, your ears, your lips, your jaw, everything, right? So your entire muscle and skeleton. What do I mean by symmetry? If you take your eyes, they will be of more or less the same shape, same size. They are almost at the same position. And in fact, when a baby is growing or an embryo is growing, they form at exactly the same time. So this we call as left-right symmetry. That is a paired organ forming with the same shape, same size, same position, etc. Right? But internally, you all know that the heart is to the left, the liver is to the right, the stomach is to the left, and so on. So they're all very asymmetrically positioned along the left-right axis. Okay. So the question that we are interested in looking at is, when an embryo is growing, how is this decision made? Right? So how do some organs decide to become singular, like the heart, liver, and so on, and move to one side of the embryo, left or right? And how do some other organs, such as eyes, ears, and some other organs, become paired and symmetric? Right? So that's the sort of overall idea of the problems that we are trying to look into. How is the symmetry as well as asymmetry is emerging during embryonic development? We use Zebrafish as a model system primarily because they're very transparent. You can look through everything that is happening within the embryos without having to dissect them. Again, Sweta and Subhanjan will show you more of it. In fact, you can study how body symmetry emerges during development in any model system. You can take zebrafish, you can take elephant, you can take humans, and so on and so forth. As I mentioned, the reason for choosing zebrafish is because it's very transparent, and the way the body symmetry or body asymmetry develops in any of these systems are very, very, very similar. So studying general principles of how symmetry develops or asymmetry develops in the body is totally extrapolatable to any other model system that you can imagine, right? So that's the primary reason why we have chosen zebrafish. Of course, it can be extrapolated to any other model system that you can think about. Okay, so that's a very brief overview. So I'll let my students take over, and they will show you the lab. They will show you how you set up fish. They will show you some embryos, some live embryos. They will show you what is symmetric. They will also show you what kind of experiments we do, what kind of analysis we do, etc., etc. So welcome again, and enjoy this next 1 hour. Thank you very much.
And one more thing for the audience. Since we can't see you, please type in your questions and comments in the YouTube chat, and we'll read your questions to the students, and we'll get back with the answers. So thank you, Dr. Sundar.
Thank you.
Thank you, Sundar, and thank you, Ullas and Arnab, for giving us this opportunity. So now we'll just show you our lab. Like right now, you're already in our lab, and we have workbenches over here where we do our experimental work.
Where's the zebrafish?
So the zebrafishes are right here. Actually, we have a whole facility with hundreds of tanks where these are housed. For this purpose, we have just brought like a couple of tanks.
Fish. So
Yeah.
can we just lift the tank and
Yeah.
bring it in front of the camera?
Yeah.
Yeah. Okay. So these, where are the fish? Yeah.
Yeah.
Okay.
We will again be showing you these in great and explaining things in greater detail. So here, this is where we do our like molecular biology experiments and so on. Over there, we have our stereo microscopes where we actually look at the zebrafish embryos and do experiments with them by looking under the scope. So yeah, and finally, we have what we call as the dark room. So the reason why we call it dark room is we have a stereo fluorescence microscope. Again, we will be explaining all of these concepts in detail, and for that purpose, we need a darkened room. And we also do most of our analysis in here in this room. Apart from this, we also have a microscope room in TRFR, which is like common to the department. We will be taking you there and showing you one of the microscopes that our lab uses extensively, which is called the light sheet microscope. So all in all, I think there's a very exciting 60 minutes ahead. So right now we will begin by explaining the question. Although Sundar did an extremely good job, we will still just...
Yeah, I would like to know, first of all, why zebrafish? What is a zebrafish? And maybe perhaps you have a slideshow?
Yes, we have that. Yeah. So, like we mentioned, we are a developmental biology lab. So, let's start off by talking about what development actually is. Now, if you look at animals around you, be it zebrafishes or be it mice, frogs, or even human beings, we all look very different in terms of our body structure, body plan, and where we live, and the kind of food we eat, and so on. But if you go back in time to the first stage of development, we're all extremmely similar, where we all began as a single cell, which is called the zygote. So now the process by which organisms move from a single cell or the zygote to such complex organisms is what development entails, and the study of it is called developmental biology. Now, again, if you look at animals around you, they are very different in terms of their shapes, their sizes, and their forms, and so on. But if you again go back, they have a very similar characteristic, where, as Sundar also explained, all of these animals can be divided into three characteristic body axis. The first one is the head-tail axis, then the front and back axis, and the left and right axis. Of this, like Sundar mentioned, the axis that we are interested is the left-right axis. And the reason behind it is, if you look at your own body plan, uh, if you look at the internal organs, there is an asymmetry in how they are positioned. For example, the heart is towards the left, the liver is towards the right, the stomach is again towards the left, and so on. But if you look at your external features, your hands, your legs, and many of your facial features like the eyes, ears, they're all very symmetric. So the question that we are interested in is, how is it that some organs form or are developing asymmetrically while there are other organs that are forming symmetrically? So the question again is, exactly how does left-right symmetry emerge during development? And the model system that we are using for this purpose is Zebrafish. Some fun facts about Zebrafish: it's called a zebrafish because it has these characteristic stripes on its body like a zebra. And it was also first isolated in India in early 1800s in the Kosi River in the then Bengal state. So, and for the last 50 to 60 years, it has been extensively used in answering different kinds of questions in development, physiology, and so on. And why zebrafish? Sundar already mentioned that these embryos are transparent. Now, what does transparency give us? It allows us to look at the entire body. Now, if you look at your own body, you cannot see the internal organs like the heart or liver or anything. And that's because you have the skin, which acts as a barrier through which light cannot pass. But now, because these embryos are transparent, you can actually look into their body, and we will show you their heartbeat, their blood flow, and so on. So that makes it very easy to look at processes that are happening inside the body. The other reason why zebrafish is a really great model organism to work with is it undergoes something called external fertilization. So what external fertilization means is the female releases the eggs outside, and that is fertilized by the males. So now you can collect these embryos and actually look at what is happening from outside. And finally, we are always interested in asking, does this have any relevance to humans? Can we extrapolate anything from zebrafish to actually what is happening in humans? And we can, because if you look at their DNA sequence, the genome, there's an 82% conservation between zebrafish and humans. So all of this makes it an excellent model organism to work with, particularly for the kind of question that we are asking. And now, like, we'll stop the presentation here and actually show you the fishes outside.
Hello, everyone. So I'll be talking more about zebrafish, like how we set up crosses, how we collect embryos, and how we store them. So yeah, in this tank, you can see two pairs of zebrafish, which are inside water. And the first thing to observe here is that the characteristic stripe-like pattern, which resembles the stripe of zebra. So that is why they are called as zebrafish. And so next, now I'll be talking about how we set up crosses. So to the day we want embryos, the previous evening we introduce a male and a female in these types of breeding tanks. So now a question arises: So how do we differentiate between male and female zebrafish? So if you observe carefully, in the tank, there are some characteristic features, like there are fishes which are more pinkish than the other. So that those are males. That is how we recognize. And for the females, the identification feature is that they have a bulged belly because of the egg pouch. So then, as I previously told you, we introduce one male and a female into these breeding tanks. The previous day we want the embryos and we let them for pheromone exchange overnight, and as soon as they receive the first shine of light, they start mating. That is, the female releases the eggs outside in the water, and they are fertilized by the male. So uh, now there is a interesting fact here, that there is a sieve we have put so that the embryos after released go and settle down and are not accessible to the fishes. So why should the embryos not be accessible to the fish? Because there is a tendency in zebrafish that they eat their own embryos. So yeah, that is why we put a barrier. So here this is a cross that was set up yesterday evening, and you can see that there are some embryos. So the dots, uh,
So the dark dots that we see at the bottom, are they the embryos?
Yes.
the transparent,
Yeah.
uh,
Maybe you can point a finger.
yeah, so here there are some. Yeah, we'll be showing you in under a microscope, then it will be more clear. So then after the embryos are laid, we collect the embryos in a petri dish, and then we store these at 28 degrees Celsius, which is the standard temperature for storing zebrafish embryos. So now you know how we set up crosses and collect embryos. Now we'll be seeing or observing these embryos under the microscope, so that I can explain the different structures in the embryo in detail. So this is a stereo microscope which is connected to a camera, and whatever we see, whatever the camera is capturing, we can see it in the screen. So these are many embryos that we have collected in the morning. So if you, I will focus at one embryo. Yeah. So now I'll be explaining what are the structures you are seeing. So this, the bigger circle or the bubble-like thing which you are seeing is called as the chorion. So chorion is the protective layer, protective structure that protects the embryo while developing. The chorion is a very important structure, because as it has already been told that the embryos are laid in the water. So while developing, they need protection from water currents and other things in the river. And then this big ball like thing which you are seeing is called the yolk. So many of you might be knowing what is a yolk. So yolk contains nutrients and other substances which help in the developing of the embryo. So now where is the embryo developing? So this structure which you are seeing on the top, like a mound, so they are the cells which have started from one cell, and then they have divided many times, and then you see a mound of cells. So this is the actual embryo which divides and ultimately lead to the formation of complex structures. So you have seen the initial.
How big is an embryo typically, size-wise?
So size-wise, it is around 700 microns, micrometers.
So not something that a human eye can see. with not bare eyes.
Yeah, we can see, but not through this detail. Yeah. So now I'll be showing you a much later stage, that is around 14 hours post fertilization. So we have collected these embryos.
First
Yeah.
one that you showed was how long after it was laid?
So this is around 2 hours post fertilization.
Okay.
Yeah, 2 hours after it has laid. So yeah, just a second, I will focus at. Yeah. So this, as I already told you, is a embryo around 14 hours post fertilization, and you can see that this is the head region, and this is the tail region, and this is the yolk. So the cells which I had explained previously have divided and then formed this type of shape. So this is the forming eye, and then if you look at this region, you can see some blocks of tissues. So this is one of the tissue of interest in our lab. This tissue is called as somites. So what are somites? Somites are precursors of muscular and skeletal system. So as it has been explained previously or told previously, that, and also we know that our skeletal and muscular system are symmetric. So this tissue becomes tissue of interest in our lab, because how these develop symmetrically and then form symmetric skeletal and muscular system later is one of the questions that we are interested in. So I will now show you a embryo in the dorsal view, the same embryo in the dorsal view. So a dorsal view means that you will be viewing the embryo from the back. So now we are viewing the back of the embryo. So this.
So, is this akin to development of spine in humans, or no? Is this akin to development of spine in humans?
Yeah. So the pipe-like structure you are seeing in the center is called the notochord, which becomes the future spinal cord and spine. And then these are the somites. So you can see that they develop on both the sides of the notochord, and it is also important to note that even while developing, they develop symmetrically. And so these eventually give rise to one vertebrae and some other muscles. And it is also known that if they do not develop symmetrically, that is, there are no equal number of somites on both the sides, it leads to something called as scoliosis, in which the patient has a bent spine or a curved stature. Next, we'll be looking at a much later stage, that is 2 days post fertilization. So basically, the embryos are 2 day old. So here you are able to see that the embryo has now attained a fish-like shape. The eyes are much more visible and developed, and this is the head region, and this is the tail part, and this is the yolk. So eventually the shape of the yolk as well as the embryo has changed, and then it is similar, it has taken a shape that is similar to a fish. So as it has already been told by Sundar, the other tissue of interest in our lab is the face. So as we see around, we know that our facial features are highly symmetric. We have the position of eyes and ears that are symmetrically placed with respect to the left right body axis. So this is called as craniofacial symmetry, and this also is a tissue or or an organ system of interest. So in this, since we cannot study all the organs, we have chosen one of the organ or tissue, that is called the palate. Palate is the roof of the mouth. So in zebrafish, it is situated somewhere in between the eyes. But yeah, in the coming slides, we'll be showing the palette in much detail. And I'll take you to other regions and interesting things that are happening in this embryo. So now, if you look at this, you see something beating, right? So, I mean, most of you would have guessed. So, this is the heart of the embryo. So, you can see it beating. So, yeah, this is why we have used this as a model organism, since we can observe the processes.
Can we zoom on the heart better?
Yeah, sure.
Okay.
Yeah, so this is why we have used this as a model organism, where we can see different processes that are happening in life, which is not possible in other organisms like mouse or even in higher organisms. And one more thing that will be interesting to see. Yeah, never mind.
We wanted to show you the circulating blood, but through
Yeah.
the camera, it's not very clear.
So actually,
So maybe
under
you can just point the mouse on that and run the mouse through it.
yeah, so here there are arteries and veins, so in which you can see the flow of the blood. But yeah, in camera it is not that visible.
Does it have a colorless blood?
Yeah, yeah. I mean, now it has, but it is actually the red blood.
It eventually
Yeah,
becomes.
yeah. Yeah, so now you have seen these fishes, and then you know they are transparent. But if we want to focus or see a tissue at better contrast, how do we do this? So for to view the tissues of interest at a better contrast, we use certain staining techniques. So one of the techniques that we use in our lab is called as in situ hybridization, where there are certain genes that are expressed at particular tissues of interest. So we stain for these genes in the tissues, and then we can visualize the tissue of interest at a better contrast.
Wait. So can we go back? So you have a technique called as in situ hybridization,
Yeah.
by which you are able to mark a tissue so
Yeah.
that it can be observed.
Yes. Yeah.
And why is it called so? In situ hybrid, is it related to some process?
Yeah. So in situ means in the embryo, in the whole embryo. We are not like taking a particular tissue or something.
So it
So
lives.
yeah, the embryo is actually dead, but still it is inside the intact embryo.
Okay.
Yeah. So I'll be explaining procedure
Okay.
in yeah. So this is the technique which I was talking about, which is in situ hybridization, where you can see that there are blocks of tissue that have been marked. So these are the same somites that I was talking to you previously. In the previous thing where I showed you, you were just able to see blocks, but it was not at a better contrast as you are seeing it here. So to perform these different staining techniques, we employ a procedure called as fixation. Since they cannot be performed on live embryos, we fix these embryos. What does fixation mean? So by using some chemicals, when we apply to these embryos, they get arrested or halted at that particular stage. Basically they are dead, but all the structures, genes, or whatever the molecules are expressed are intact. So this is called as fixation, and then we do the different staining techniques so that we can observe the tissues of interest at that stage.
So
Yeah.
during the developmental stage,
Yeah.
you are able to see at whichever stage you are interested in preferentially by
Yeah.
arresting it and staining it.
Yeah.
It's like preserving so that you can view it immediately.
Yeah. It is similar to, I mean, people would have seen how the specimens are preserved in formalin for many years, right? So this is similar to it, where the structure is preserved, and we can also stain the.
So it's like the kids play statue.
Yeah,
You just
exactly.
praise it
Exactly.
and then see it.
Yeah, exactly. And now I'll be showing a different embryo where we have perturbed a gene, blocked a gene, and then we see asymmetry or different number of somites on left and right side. So this is the left side of the embryo, this is the right side of the embryo. So you can see that here there are more number of somites with respect to the other side. So yeah, this is the kind of asymmetry that I was talking about. So it is shown that these these types where the somites are asymmetric on.
Just one.
Yeah.
Because when you explained that, it was not very visible on
Oh,
the
sorry.
camera. So can you go back a minute
Yeah.
earlier?
Yeah. So this is the left side of the embryo. This is the right side of the embryo. So you can see that on the left side, there are more number of somites that have been developed and stained with respect to the right side. So this is the asymmetry which I was talking about. And it is, as I have already mentioned, that these kinds of asymmetries leads to a condition called as scoliosis. If scoliosis, that is, the people have a bent spine or a curved stature. Yeah. So, and now Shweta will be explaining another staining technique which we use in the lab.
Yeah. So far, Subhanjan has shown you zebrafish embryos at different stages, and he's also explained one kind of staining technique that we do, where we look at the somites by a technique called in situ hybridization. Now, like he said, the lab is also interested in looking at craniofacial symmetry, or what it means is like how are these facial features in our body developing symmetrically. And as he mentioned, the tissue that we are interested in is called the palate. So what is the palate? If you take your tongue and take it to the top of your mouth, you would hit a barrier. and that barrier is the palate. It's the roof of your mouth. So we look at this because it's also there are also syndromes associated with incorrect development of the palate, which is called cleft palate, and it is a very I mean, it's quite common in India as well, with an incidence of 1 in 1,000 babies having this syndrome called cleft palate. Now, how do we look at the palate? Now, this is a complex problem for us in the lab, because if you look at where the palate is, you have the brain in your head, you have the eyes, which are quite huge in case of zebrafish, and underneath the eyes you have the palate. So looking at it without any kind of staining is almost impossible in the zebrafish embryo. So we employ a staining called Alcian blue staining. Now, I'll show you how, I mean, why it's called Alcian blue staining, simply because the stain looks blue. But what it stains for is the cartilages that are present in the embryo. So, cartilage is a kind of soft connective tissue which is present in your ears, the pinnae of your ears, in your nose, and so on. So the palate is also a cartilaginous tissue, and this stain binds to all these kinds of cartilages, allowing us to look at it. So here you can see that actually the larvae is also stained blue, but there are certain structures in the larvae's head which are stained darker in blue. And these are the cartilages that I was talking about. Now, I also have two samples where I have just the head and, sorry. So this is the zebrafish head looked from the bottom. So what you're seeing at, or what you're looking at is the lower jaw of the zebrafish, and you can see these several rod-like cartilages, which are called the Meckel's cartilage in zebrafish. And again, this is the zebrafish head from the top. But now, again, it's not easy for us to directly look at the palate, because there are simply too many cartilages that are present in the zebrafish head. So what we do in the lab is a process called dissection, by which we carefully remove out the brain tissues, the eyes, and the lower jaw, and finally we come up with a tissue that looks like this. So
when you do this, roughly after how many hours of embryos development you pull? Like 15 hours thing was.
Yeah, so the zebrafish palate begins to form at 13 hours post-fertilization. So these arise from a special cells called neural crest cells, and these neural crest cells then reach the face, and the final palate is actually formed at 56 hours post-fertilization, so roughly between 2 and 3 days. And what we have over here is a 5-day-old larva's palate. So at 5-day-old, it becomes completely cartilaginous, which is why we are able to do this.
After 5 days.
Yeah. If by 4 days you already begin to see it, but by 5 days it's very clear. So what you see over here, this is the zebrafish palate. So it has this plate-like structure, which is called an ethmoid plate, and these two rod-like structures. And you can see it's a very symmetric tissue. Like if you can draw a midline between this, and the left and right would be very comparable. So there are 3 people in our lab who are looking at how this symmetry emerges right from, and they're looking at development right from 13 hours until 5 days to see different stages of palate development in the lab. So this is one of our very exciting projects in the lab. But now we'll slightly shift gears, because whatever Subhanjan and I have shown you so far, it's it's like we are arresting development and then we are looking at the formed tissue and assessing for whether it's symmetric or not symmetric. But we are also interested in asking the question of how does symmetry emerges. So for that, we need to look at live organisms and not just do this process called fixation and look at tissues by staining. And the other thing is all of these techniques are at the tissue level. We are looking at somites, which is a tissue, or the palate, which is a tissue. We really need to understand things at a cellular scale, because cells are the fundamental units that makes up all of these tissues, and we want to understand how these cells are behaving within live embryos. So for that, we do a technique called fluorescence microscopy in live embryos, and Subhanshan will start off by explaining what fluorescence is, and then we will take you downstairs to our light sheet microscope room.
There is one YouTube question.
Yeah.
Is the brain also has the similar kind of zebrafish brain also has a similar type of symmetry as of humans? And do you do research on this brain symmetry?
We are not looking at brain symmetry directly, but so the brain does have a left and a right half, and it also has a midline. But the functionalization in the brain seems to be very different between the left and right halves. So, I mean, that is a very interesting question by itself. We are at this point not directly working on it. Yeah.
One part of the question was, is the brain symmetric left to right?
At
At least in zebrafish.
Uh,
Or you're not really looking at it?
We're not directly looking at it. I mean, if you look at just the tissue structure,
Yeah,
it does
from the
appear.
tissue structure
Yes.
point of view,
Yes, it does appear.
it is symmetric.
Yeah, we have not quantified or made measurements to that, but it appears.
It appears to be symmetric.
So now Shweta mentioned a phenomena called as fluorescence. So what is fluorescence? I mean, some of you at least might have heard the term fluorescence, or somewhere seen movies or something as well. So fluorescence is a phenomena in which there are some molecules which, when are irradiated by a particular wavelength of light, they absorb the wavelength and then they emit a higher wavelength. So this is called as fluorescence. But how do we introduce this fluorescence in the tissues or cells of our interest? So in around 1960s, there was a major breakthrough in which a laboratory called as Marine Biological Laboratory. So scientists in there found out a jellyfish which actually glowed. So
At the bottom.
yeah, at the bottom of the sea, and they were interested like how is it glowing at the bottom of the sea. So then after further research, they got to know and they discovered that there is a protein which is a biological molecule and biomolecule and present inside all the cells. So a particular kind of protein is present in the jellyfish that glows. So but when irradiated by a particular wavelength of light, that is why the jellyfish was glowing. So since it also happens in biological tissues, so what the scientists did was that they extracted the gene of the protein that produced this GFP and or green fluorescent protein, and then they introduced into different organisms, like they introduced it into bacteria, and now we use the same technique and then introduce the gene at the back of the gene of our interest that is expressed at the particular tissue of our interest. So what is the ultimate result is that whenever we irradiate the embryo with the particular wavelength of light, only that tissue absorbs the light because that has the protein or the molecule which has the phenomena of fluorescence, and then only that part gets irradiated. So this is the phenomena of fluorescence. So here I have a vial where we have this green fluorescent protein which have been extracted from the bacteria. So
Can we turn off the lights? Yeah.
last switch. So this is a UV torch. So whenever we irradiate this,
So let's reduce the light further. Or maybe you come to this problem. One, you come to
Yeah.
this problem.
So whenever we shine UV light against this,
Yeah.
you can see that it is glowing. Isn't it mind-blowing?
So can we turn off the light? And then there is no illumination.
No
Nothing,
illumination.
no light comes out of the
Yeah.
sample.
And we can also do experiment where we can just shine a white light or any other light and it does not glow. It only glows when it is irradiated at 488 nanometers.
So you are shining
A UV light.
UV light on it.
Yeah.
Yeah. Okay. Good. Good. Okay. Can we have the lights again? Yes.
So now we have some transgenic lines where we have introduced this GFP and other fluorescent proteins into the tissue of interest, and that is how we can observe the tissues carefully and at a better resolution or even at the cellular scale in the microscope. So now, as Shweta mentioned you that live imaging or it is important to know how the symmetry emerges when the embryos are alive. So now for that we employ a technique called as light sheet microscopy, which uses the same fluorescence technique. So for that we'll be going to a microscopy room where
So when we walk to some other room, so Shubhanjan, would you like to tell us exactly where did you start your research journey and what attracted you to this field?
Yeah. So I did my so even after my 12th, I was quite sure I will
So
go.
where are you from, by the way?
I'm from Bangalore. So I did my schooling 11th and 12th, even graduation in Bangalore.
So you're born and brought up in Bangalore.
Yeah, yeah. So then uh, even after during my 11th and 12th, there were some teachers which helped me to get interest in research. So that is how I chose pure science. So then I pursued bachelor's uh, applied to many places, but then this was one of the lab where
So you
I got
wrote the
interested.
famous JJ Bills.
Yeah.
And then you had a couple of rounds of interviews.
Yes.
And how was it? What was
I
your
mean,
experience like? Was it all fun?
Yeah, it is a great experience. Especially the interview in TFR Mumbai and other TFRs. The experience itself is great, though you get in or not. But just the experience is itself great. So then after that, I was interested in several labs, but I was placed in this lab, which was also one of my choice.
So you were given a choice of two laboratories.
Four
And
laboratories.
they were to like select one of them.
Yeah. Since I am an integrated PhD, that is the case.
So though professionally you have varied interests and you would like to do many more things.
Yeah.
Okay. Sounds interesting.
For PhDs, you have rotation and so on.
Okay. So hey, you have lots of microscopes here.
I just gave.
Yeah, so we have 3 microscopes in this room. This is a confocal multiphoton microscope. I know I'm using a lot of
Okay, but you're not going to show us this instrument. You're not going to show us that either. And you're going to show us this one. Okay.
Yeah, this is the microscope that we in our lab work with a lot. So this is called a light sheet microscope. I'll explain why. Yeah, I'll explain why it is called a light sheet microscope in just 1 minute. So yeah. So as Subhanjan said, the principle of fluorescence involves excitation of a fluorophore, that is a molecule that can fluoresce by a wavelength of light, and it then upon excitation by this light, the fluorophore emits a light with a different or higher wavelength. So now, to illuminate molecules with very particular wavelengths of light, we use lasers. Lasers are sources of light which can be composed of just one wavelength of light, allowing us to excite fluorophores very specifically. And the way the light sheet system works is you have two illumination sources. So you have one on the left and one on the right. And both of these illuminations, or what we call as illumination objectives, generate a sheet of light which strikes the sample, and hence we call it a light sheet microscope. And the light then from the sample is collected by a detector which is placed perpendicular to the direction of illumination. So now,
Is it like you're having light in a single plane?
yes,
Shining on sample?
yes.
And any specific reason for this?
So one reason for it is you want to collect signals from the sample plane by plane. Otherwise, there will be a lot of background that is coming from different planes. One way where people do it is to use a confocal microscope. So a confocal microscope are the other kinds of microscopes that are there in this room. And what it has is a pinhole which collects light from every point on a single plane, and then it moves in a 3D axis. Now, as you can imagine, if I'm moving across a grid point by point, it will take me a lot of time to go across the entire grid and then go in the three-dimensional depth of the sample. So the light sheet saves us a lot of time because it's illuminating an entire plane at the same time, and also just illuminating a specific plane. So it combines a lot of benefits by which we are able to look at the structures at a better resolution with less background.
And you are able to rotate the plane as it is.
Yes, yes. I will show you how that works in the system. Like, we actually will be able to rotate the entire sample and get a three-dimensional view as well. Yes.
Because you are getting essentially by looking at different planes at different times, you are getting a 3D picture.
Yes, yes. Okay. Yeah. So we can move to the scope right now. So the part that I showed you in the picture with the illumination and the detection objective is here. So you have a left illumination objective, a right illumination objective. This is the sample chamber. So this is where the sample will come, and we load the sample from the top over here. So, so we load the sample in something like this. So this is a glass capillary, which is held by this holder.
Very visible.
Yeah, this would work.
Yeah.
Yeah, so we hold the capillary in a sample.
Larva is in the capillary.
Yeah, the embryo is over here. Yeah. And so the way the embryo is mounted is in agarose. So I'll explain why we use agarose. Before that, I'll just show you the detection objective, because it's lying behind the sample chamber. So from this view, what you are able to see is the sample chamber, and behind it you see the detection objective. It's not very clear now, and the reason for it is the sample chamber needs to be filled with water. Now, why water? So if you have ever put a stick or a pen inside water, you would see that the stick appears bent when it's inside the water, right? That's because of a phenomenon called refraction, which is the bending of light. Now, when we capture images, we capture it in live embryos, which are in a buffer which is very similar to water. So we use all the optics in this system at a refractive index which is similar to water. And hence, the moment I fill this chamber with water, you would see that the objective has become very clear. And that's also why we mount these samples in agarose. So agarose is a jelly-like substance in which the sample is stable, but it also has a refractive index which is very similar to water. So now, when I
This is basically to eliminate the error due to refraction of light.
yeah. So everything, so the refractive index of all the lenses, the illumination optics, the detection optics, the chamber, everything is set to the refractive index of water 1.33, so that we can fill the chamber with water and image the embryo in a live condition where it can grow properly. So that's how we work with the system here. And now, like I said, the sample comes from the top. So I'm just bringing it down over here. And you can see that it's there. And what we do next is remove the sample from the glass, because glass has a different refractive index. We just need the agarose. And you can see that despite the sample being inside water, it's held stably by agarose. And now I'll show you how it looks like when we illuminate it with a wavelength of light using lasers. And in a fluorophore which marks all the nuclei in the embryo. So here you can see the zebrafish embryo, which is arrested in development at early somite stage. And these are the somites. It might not be very clear now because these are just the nuclei. We also have a fluorophore over here, which marks the cell boundaries. And now you can see the somites quite clearly. So we can also zoom into the embryo slightly more. And here you can clearly see the somites on the left and right side. This is the notochord, as Suvanjan mentioned. This gives rise to part of the vertebral column, the intervertebral disc. And somites essentially ultimately give rise to the musculoskeletal system. They give rise to the ribs on both sides. So each somite crudely becomes one pair of ribs. And they also give rise to part of the intervertebral disc and many of the skeletal muscles as well. So this is just one snapshot that I have shown you over here. Like this, we are able to image live embryos for hours together to see how development actually happens. And now I'll be moving back to our presentation to show you some of the movies that we have acquired in this system. And yeah. Yeah. So on the left, you again see a somite stage embryo, where right now the embryo is lying parallel to the ground. So what you see in the middle is the notochord, and on either sides you see somites developing. And the question that I am asking in the lab is, how does a somite gain its final shape, and what happens, what are the mechanisms that are regulating this process? Similar to the somites, we've also been able to image the palate. So the palate over here is just forming. And so this is the rod-like structure that I showed you earlier. And this is the plate that eventually forms. So initially it's not completely fused, but later the palate forms over time. So people in the lab are also asking how this palette is forming, how the fusion is happening, and so on. So finally, we will move to why are we looking at symmetry? Like it's
This slide here.
no, it's there on the slide. Yeah. So I mean, it's obviously a very interesting question to ask because, like, when you have certain organs developing asymmetrically and certain organs developing symmetrically, how does this happen during development? This is a very interesting question to ask, but it also has very much of a clinical significance as well. Subanjan mentioned multiple times, when you have asymmetries in somite development, it can lead to a condition called scoliosis, where you can end up with, where people having this condition have a curved or a bent spine. So this is how it looks like. And in zebrafish, people have been able to come up with models and perturbations which can mimic this condition. Now, by looking at what is happening in a zebrafish scoliosis condition, we might be able to come up with mechanisms or identify how it is happening in humans, and over time even come up with cures for this same.
Let's hope to find the origin of this disorder.
Yeah.
Okay.
And like I mentioned earlier as well, so the palette forms by two streams fusing at the region where it eventually forms. Now, if this fusion does not happen properly, you end up with a condition called cleft palate, where you have a gap in the palate. And sometimes babies can have a syndrome of both cleft palate and lip, where you have a gap both in the palate and the lip. And this is a fairly like it has a decent or sadly it occurs in 1 in 1,000 of babies in India. So again, there are zebrafish model organisms where you end up with very similar characteristics, where there are gaps in the palate. And again, we hope to uncover the mechanisms or why this happens within zebrafishes. Finally, if you're interested to understand more about left-right symmetry or understand more about development in general, these are some of the references for which you can look up for. So the developmental biology textbook by Gilbert is one of the standard ones across colleges, which is definitely a very interesting read for biologists who are interested in this. And our lab also has a review and article on emergence of left-right symmetric body plan in different tissues. So that is it from us. This is our lab, and if you have any questions or if you want to read more about the lab in general, you can visit our webpage or email us at any point, and we would be happy to get in touch with you and talk more with you as well. Yeah.
Okay, so can we just check if there are questions?
Yeah, is there any particular fish which is similar to zebrafish, which can be transparent? Is one question posed.
Yes.
Sir.
So there are fishes called medaka, which is also being used not quite extensively as zebrafish, but they are also used in research as well. So that's definitely one model organism. Yeah.
I have one question. What is the first organ to be developed in a zebrafish?
So the first thing that happens across species is the specification of germ layers. So what I mean by germ layers is all our tissues arise from three germ layers, which is called the ectoderm, endoderm, and mesoderm. So this is the first specification to happen. And in vertebrates, the first tissue that forms is the notochord. The notochord is actually, in a way, a transient structure because it forms part of the vertebral column, and that's it. So, but that is the first in case of vertebrates, yeah.
That's it from YouTube.
Mhm.
That's if you have some questions.
Maybe you would like to tell us, like Shubhanjan explained, what got you interested in this area of biology? What triggered your interest?
So the major fascination for me came in 2019. This is a shout out to ICTS's quantitative systems biology workshop. So this was a 3-week school where they had introduced us to morphogenesis or like developmental biology. So they had physicists, they had biologists, all of them who were working on different questions in development. And this was the first spark for me. And when I met Sundar over here, he is he's a fantastic PI to work with. So that the question that he asked, and this also got me interested in this. Yeah.
There is one more question from YouTube.
Yeah.
It's from Monali. He's asking how cells are self-aware of when to stop growth. Okay. And to which extent it should grow, and what is the factor it decide of when to stop?
So actually, that's very close to the question that I'm asking in the lab. So we don't know entirely what tells a cell to stop growing. We think that so there are a lot of chemicals called morphogens that regulate development. So we think that removal of these morphogens over time is one of the things that tells a cell or a tissue to stop growing. Or there can also be some because during development, there's a lot of cell movement that happens to form very specific shapes. We also think there might be some change in the tissue material property. Like what I mean by material property is whether it's fluid or solid and so on. So we know that solids don't change their shape easily, but a fluid can change its shape very easily. So we think that there might be transitions like this that is also happening during development, which tells a cell or a tissue to stop growing or stop changing shape at this point. Yeah.
The three fundamental questions: why, how, and when.
Yeah. And we definitely hope to uncover more of these answers in the next few years.
So
Yeah.
I also have another question. You are specifically dealing with disorders in zebrafish, a particular kind of disorder. Is that a natural disorder, or you make the fish to
Are you able to trigger the disorder?
We are, you can
Yeah.
answer that.
So we are able to trigger the disorders. So we have found some genes which are involved in it. So as you asked, these disorders are naturally occurring. So we have made those models in the zebrafish as well, so that we can study how does it happen. Since it is not ethical and we cannot study it in human embryos or the other embryos. So now we use zebrafish as a model organism to study how are these happening. But in our lab, we are not actually concentrating on the disorder itself. We want to ask fundamentally how is it developing symmetrically, how the symmetry emerges. And since if we know the mechanism, then we can also answer like how are the disorders occurring. So what is getting perturbed or what is getting harmed during development, that is leading to that disorder. So
very interesting.
Yeah.
So on this note, I think if there are no more questions, it's time to thank today's presenters. And Dr. Sundar had to proceed to some other place with some other work. So right now he's not with us. So thank you, Shubhanjan, and thank you, Shweta. And on this
people
note,
also want to visit the lab. They are requesting for lab visits.
okay, so for lab visits, please write to outreach@tifr.res.in. Lab visits especially to biology labs in TIFR are very unlikely to happen. And the institute is open for visit by general public during National Science Day celebrations, typically towards end of February. So people are welcome to visit the institute around that time. Meanwhile, keep watching Chai and lab pages and different web pages of TIFR. And there is a lot of interesting stuff happening, and we'll be posting new data as and when possible. So on this note, thank you very much.
Thank you.
Bye-bye.
thank you for the opportunity.
Yeah.
Great. Good.